Hello, everybody. My name is Lydia Poplinsky, and I'm doing my research article presentation on the recent pandemic virus, which is SARS-CoV-2, more specifically looking into the ACE2 binding of dalbavancin and the effective inhibition of this antiviral protein. At the time the paper was written, there wasn't a vaccine already made. So the major goal of this paper was to continue the important research of finding a vaccine for the global pandemic that has just caused so many deaths. So the virus is a non-segmented enveloped single-stranded RNA. Viruses typically inject their DNA or RNA into a particular cell and overtake that cell's mechanisms. That can cause certain symptoms within us, and it can cause death and other issues. A really important part of the COVID vaccine are the spike proteins located on the outside of the virus. These spike proteins can bind to a receptor and then inject their cell. Specifically with SARS-CoV-2, they bind to ACE2 receptors. So in order to get a vaccine, the goal is to find a competitive inhibitor to locate all of these different ACE2 binding sites so that SARS-CoV-2 can no longer bind and can no longer affect us. This is a picture of ACE2 as well as the glycoprotein spike. The last thing I want to indicate that is important is there is a similarity to the virus SARS-CoV, which is different from SARS-CoV-2. So they have an 82% sequence resemblance, as well as receptor binding domains that are conserved. And the importance of this is that they were to infer a lot of the SARS-CoV-2 interactions based off of the SARS-CoV already present knowledge. And so this was really helpful. They knew that SARS-CoV-2 likely binded to ACE2. So with this knowledge, they wanted to find an antiviral agent that was able to competitively bind. And so they predicted that dalbavancin administration would reduce the viral loads and ultimately result in SARS-CoV-2 infection no longer binding or no longer happening because all of the dalbavancin would be binding to this ACE2 protein site and there would be no space for SARS-CoV-2. There isn't a lot of time in this presentation, which is totally okay, but there's a lot of methods that they use because they're trying to prove in as many ways as possible that dalbavancin is an effective drug for treating SARS-CoV-2. So in an effort to help you guys understand all of the results that they have, I wanted to describe a little bit about some of the methods. So the first one is QRT-PCR. Some of you might have seen PCR already. The RT just stands for reverse transcription. And this is a way of amplifying DNA, and it can help you look further into DNA detection of genes and a lot of other analysis. The next important assay is an ELISA. And ELISA uses an antigen and several antibodies to identify this antigen-antibody binding connection. So this is important for us because the ACE2 is the binding site, and SARS-CoV wants to bind to that as well as dalbavancin. So we'll see a lot of ELISAs that prove the quantity of these binding sites. The last one is Western blotting, and this one is a really helpful tool for identifying whether those antigen-antibody bindings are present. The first step for this research is to identify that dalbavancin is the most efficient antiviral agent. So first, they went ahead and went to the FDA-approved peptide drug library. They found 10 candidates that would totally inhibit ACE2, and they did an ELISA and a co-immunoprecipitation to figure out that dalbavancin seemed to show the strongest inhibitory ability. Here is a dynamic simulation of ACE2. This red circle indicates the binding site of ACE2 with SARS-CoV-2, And once we move over to here, we'll see a number of different arrows. The red and blue arrows show high charge areas. So those are the interpolated charge surfaces. And then the green and purple arrows indicate and show hydrophobic domains. Then you have this middle site that is kind of showing you the importance of these amino acid residues and the relationship between dalbavancin here binding with ACE2. This last section right here shows different sort of interactions, things like London dispersion forces that are very important as well. The next step to their research is to, one, prove that dalbavancin binds to ACE2 and that it binds well and it inhibits SARS-CoV-2. So briefly, I will describe each of these figures as there is a lot to them, but we don't have a lot of time. So NA is a Western blot performance or assay that they went ahead and did. This is a number of different different antigens that they think may have an ability to inhibit SARS-CoV-2 from binding to ACE2. So each of these show a different concentration of ACE2 binding successfully to the SARS-CoV-2. So you can see the most promising are dalbavancin, octaglutatone, and polymyxin B. A similar assay was done with B, but it was just done with a different group of cells. And so they got a little bit of different results. And then C shows SARS-CoV-2 binding to ACE2. So as you increase the concentration of SARS-CoV-2, there is more of a response unit, meaning that there is more binding. There is more product of both the SARS-CoV and the ACE2 binding. And then dalbavancin, you see a very similar pattern. The results are now also analyzed between how competitive is this inhibitor. So yes, it binds to ACE2, but does it want to bind to ACE2 more than SARS-CoV-2 does? So groups A and B here in this figure are just showing that exact trend, that the more dalbavancin you have, the more inhibition of SARS-CoV-2 there is. It also proves the cytotoxicity is not bad. It's really good, actually. There's not a lot of cells that are dying. In both A and B, these are done in different cells, as well as C. Similar assays doing PCR, counting cells, going through incubation, doing co-immunoprecipitation, which is very similar to ELISA's. The next step in letter D, it helps us show that the dalbavancin was the most effective if introduced before and after SARS-CoV-2 entry. And then looking lastly at F, it's really important to understand the amino acid residues and how they interact in that binding site of ACE2. So what they did here is they changed a number of different amino acids located on that ACE2 and saw if that changed the dalbavancin's ability to bind. Because if it did, if this bar is lower, that tells us that this amino acid is really important to that binding site. So in conclusion, 2 through 6 and 10 and 11 were really important amino acids. So we're just getting a better understanding of the general structure and the general effectivity of dalbavancin. Now they tried the dalbavancin on mice models. And so this first graph right here is just showing that there's less of a viral load with the dalbavancin present than without, which shows that the dalbavancin is successfully inhibiting SARS-CoV-2 from binding. So this is also seen here in B. The little pink and purple sort of sections that we see here are lymphocytes and macrophages. And those come when there's an infection, when there's something going wrong. And so we see a lot more of infection without dalbavancin present than we do with it. And so it is really awesome because this is one of the first steps showing that dalbavancin is effectively treating the coronavirus. In fact, there is 10 to the 7th more without dalbavancin than 10 to the 3rd with dalbavancin. So it really does slow down that viral load, which is what their goal was. Lastly, the dalbavancin was analyzed in these monkey models. And so the first step is, okay, we're going to create a control, which is the saline. We're going to create the dalbavancin group. And so these ones were treated with dalbavancin, and all these different tests were taken. And again, we don't have quite enough time to talk through all of it, but you can see and see here, these circles represent pulmonary infiltration. So this just represents very dense areas. You have a lot of visible pathological changes, and even the histological analysis that we saw on the last page is also present here. You see a lot more pink in this left side, in the control side, without dalbavancin present. In B as well, there's this indication that dalbavancin has a long half-life, which is really important when you're looking at virus specific ways to just stop the virus from continuing to spread. You want something that has a long half-life. So in conclusion, the article showed that dalbavancin was a really promising agent in stopping SARS-CoV-2. So it effectively prevented entry of SARS-CoV-2. We also learned some important amino acids involved in this process, and ultimately showed that dalbavancin is a promising antiviral agent through many, many different assays. Thank you for listening to my presentation, and that is all.